Hi, I'm Binita.
Hi, I'm Shumon.
Today we have with us Dr. Benjamin Martin. Ben was born in Vancouver, British Columbia. Ben was a part of international hockey from Canadian team for 2016 Olympic in Rio de Janeiro. He completed his bachelor's in biochemistry and molecular biology from University of British Columbia. Later, he joined PhD in the same department and worked with Dr. Howie Lian.
Ben has received several awards, which includes Best Doctoral Thesis Award. Also, he had twice won Alexander Graham Bell Award, hosted by Natural Sciences and Engineering Research Council of Canada. He was a postdoctoral fellow in University of British Columbia, and currently he is doing his postdoctoral research in Edelman in Harvard Medical School, Department of Biological Chemistry and Molecular Pharmacology. Ben will give us a short presentation of his journey and work.
I'm really excited for our discussion. For this part, the presentation part, I'm just going to give you a kind of a big picture overview of my research field. Kind of what are some of the big questions I'm trying to understand, and what do I hope to spend my career, what type of questions do I spend my career trying to answer? So we won't get too much into the nuts and bolts, but we'll just stay big picture. And so kind of one of the central questions is the control of gene expression. This is central to most cellular processes and is especially important for cell state transitions and differentiation. Fundamentally, each cell in your body contains the same genetic information, the same DNA. But from that one set of instructions, you have many different types of cells. And that's because they're using that DNA differently. So you have different genes being expressed in these different cell types. And a really important question of that is specificity. So how are the right genes at the right time being turned on or off? And my overarching goal is to understand how is that regulated? How are genes activated or repressed? And to understand this question, I want to dive right into the mechanism by which gene activation occurs. And to understand that, we got to understand, okay, what does the genome look like? And so with some notable exceptions, like the dinoflagellates, all eukaryotes package their genome with histone proteins into a nuclear protein structure called chromatin. And that's what you see here depicted on the slide. And so at its core, you have 8 histone proteins come together to form an octamer. And this is wrapped by about 147 base pairs of DNA. And this is called the nucleosome. Chromosomes are then packaged into many, many nucleosomes, and these form higher order structures with some regions of DNA being more or less accessible. To give you an idea of the scale of this packaging, a normal diploid human cell contains approximately 30 million nucleosomes. And so that's 30 million nucleosomes in every single cell in your body. So you can think about how many nucleosomes one person contains, and it's a lot. But chromatin is not a static entity. And so this nucleosome structure can be dynamically modified. The nucleosomes, where they are, they can be moved, they can be removed, and they are modified in all sorts of ways. One area that's heavily studied is the chemical post-translational modification of the histones. So we can add little chemical groups onto them, often on these outer bits, the tails of the histones, and these can be added and removed. And that changes the properties of chromatin. And it's thought to be all tied up with how genes are turned on and off. And so much of my research is trying to understand the relationship between these histone modifications and changes to the nucleosome and gene expression. So how can chromatin modifications promote or repress gene expression? And conversely, how does this process of activating genes alter the chromatin of those genes? The key player in this process is the enzyme that transcribes genes into messenger RNA, which is RNA polymerase II. And so what I'm going to show you here is just a very short video. This was a beautiful structure. So this is cryo-EM data solved by Lucas Farning when he was in Patrick Kramer's lab. And RNA polymerase is here shown in gray. And it's reading the DNA that's in blue. And you can even see the start of the RNA being transcribed in red. In front of it is the nucleosome. which forms a major barrier to RNA call 2 progression because it's a very stable structure and it restricts the ability of the polymerase to move along the DNA. And so something I'm trying to understand is how does polymerase navigate this obstacle and what factors can both facilitate these chromatin transactions and maintain nucleosome integrity. On the top of the nucleosome, you can see this structure there. Kind of looks like a unicycle on the nucleosome, and that's a factor called FACT, which is one of the things that I've actually studied. And so that's the big overview of what I spend my time trying to understand. I'll bring it back to this idea of specificity, which I think is so interesting. There's so much we don't know yet. So there's lots of questions, and hopefully questions that I will spend the next however many years of my life trying to answer and understand. So thanks again for having me. I'm looking forward to our discussion now.
Thank you so much for that interesting talk.
Everybody has a story that how they found out that science was the thing for them. How did you fall in love with science?
Yeah, yeah, that's a great question. I mean, I think everyone has an interesting story for how they found themselves in science, what got them excited initially. For me, it was actually during my undergrad. It was in the last couple of years of my undergrad when I started to do the upper level lab. So it was one lab course taught by someone called Scott Covey that was really influential on me. And it was the first lab I'd done where rather than being structured as here, do this experiment and get this result, it was structured as think about the questions that we're trying to get at. Here's how you try and answer them. And it wasn't a given what the result would be. And so it was starting to make that transition from learning to discovering. And then from there, I went on to do an undergrad thesis project in Leanne Howe's lab where I did my PhD. And that was where I really got given a question about biology and told, we don't know the answer to this question. Can you do some experiments and try and answer it? And to be stepping into the unknown and trying to answer an unknown, like a question we don't know the answer to, was really exciting. And that's when I fell in love with science, and I really haven't looked back since.
Wow, that was amazing.
Being a professional sports person is a dream to many. But often it is either you choose a sports or you choose another profession like academics or being a researcher. We would like to hear your journey from being an international hockey player to being a scientist.
I kind of stumbled into both, or I always wanted to play hockey. And when I was able to play hockey in my undergrad for the University of British Columbia, and it wasn't until near the end of my undergrad that I started to get some opportunities with Canada. And so that was kind of my focus and my goal. And then as I was doing, so it was actually while I was in that undergrad course with Scott Covey that I mentioned, that was when I got picked for my first tour with the Canadian team. So I went and played a training tour in the US, and then I went to the Pan Am Cup in Santiago in Chile. And so it was like everything was really happening at once for me there. And it was about 6-7 months later that I did the undergrad thesis with Leanne. And I think sometimes ignorance is great, and like the confidence of youth to just think that things can just happen. And so I was fortunate in that lots of people gave me help. And so like Scott Covey, for instance, was like, okay, you're gone for a month and you're missing all this junk, but we'll just make it up when you get back, or we'll kind of adjust what you need to do. And so right away, I kind of had this, I was lucky to find people who were able to help me balance the two. But really what it came down to is I was just very excited about doing science. And I thought that, oh, why not try and do both? And so basically and then it was about a year after that first tour a year and a half that I started grad school. And so then grad school is always in the context of hockey. And Leanne, my supervisor, was just an amazing supportive mentor. in that I would go to her and be like, Leanne, I've been picked again. I need to leave for 3 weeks to Malaysia or somewhere. And she would always say yes, and she'd often watch the games if there was a stream and email me about them. And so, yeah, just so the two together just kind of happened.
That's really amazing. Having people to support you is something huge.
What was the most challenging phase of your career?
Yeah, I was thinking about this. I probably got two answers. There's one very specific time that was really challenging, which was the first time that I had a really scathing review with a paper rejected. And so the peer review process, you send off your paper to a journal, and 3 anonymous reviewers read it and give their feedback, and then the editor says, whatever they say. In this case, it was rejection, and you get to see their reviews. And one of them especially was the person, they hated the paper, that it happens, but they were quite scathing in their reviews, and so it was really discouraging, and so that was a tough time to get through. So that's one answer. I think the other answer is there were probably some years when I was doing hockey and grad school that were really tough. When I was, we had like an intense training block where I was in town, but we're training. We were doing like 6 a.m. practices, and then like afternoon sessions, and I was going to lab in between. And so it was just, there were some stretches there where it was just, I was very tired all the time, and that was challenging.
That's really inspiring, Ben.
One thing I did find, like even though balancing sport and field hockey and grad school was really tough at times, in some ways it was really beneficial. For both sport and science, you have ups and downs. And sometimes it can be tough to get, if you have like a long stretch where things aren't working, or with sport, if I had a bad run of form and I just things weren't clicking, mentally, I found it so good to have some other endeavor that I cared about a lot. And so if things weren't working on the field, I could go and do some experiments and forget about it and just be completely engrossed in a presentation or whatever else, and then come back to hockey with a fresh mindset. And then probably more often the other way, if I was going through a tricky patch in the lab, you're optimizing something that's just not working, or you've got like your favorite hypothesis and the experiment that you've held on to for years, and then the experiment comes back and it's wrong. Being then able to go and just play hockey and completely forget about it was so helpful for me. And so that's something I try and keep. Like now that I'm not playing hockey, I try and find things in my life that I'm emotionally invested in, and that I can just go and do and not think about science for a while.
So on that note, what do you love to do most when you're taking a break from science? Yeah.
So I still do play hockey. I'm on a club called the Minutemen here in Boston. And so having that outlet is really good. It's now more social than competitive, but still a lot of fun. With my wife, we really love hiking and kayaking and just getting outside. And so, we try and get out and have some good hikes. We've been exploring, because I'm in Boston now for my postdoc, we've been exploring kind of the local mountains that they have here and the coast, and getting outside in nature is high on my list.
He also writes haiku poems, which you can find in his Twitter format in haiku.
We'll also put some links in the description box so that people can find your poems.
Being guided by excellent mentors and also mentoring a lot of students as a co-supervisor in University of British Columbia, as well as now in Harvard Medical School, what best and worst mentoring advice have you come across?
Good question. I'll circle back to best. I'll start with worst. And rather than giving like a specific advice, I'll give a category of advice that I think it's easy to fall into, and is not very helpful. And that's, I often see people, and catch myself as well doing this, of basically telling their life story in the form of advice. And so as an example, I might tell you that to become a better scientist, you have to receive a scathing and personal review when an article is rejected, because that happened to me. And I might look at my life and see how I think that made me a better scientist. I might believe that. It might be true. I doubt it. I think that I would be just fine without that experience. I think it was just a pleasant thing that I went through. But the thing is, even if it did help me, even if that was part of my story and that helped shape me, that doesn't mean it will help you. And so look out for autobiographical mentoring advice and see it for what it is, which is it's an N of one. This is someone's story. It may have helped them, it may not. It doesn't necessarily mean it will help you. The term that you see thrown around, one of the terms that's thrown around is survivorship bias. And so you're just hearing people who made it through, and the things that happened to them aren't necessarily the things that helped them get through. So I think that's something I look for in myself. I think it's hardest to see it in yourself when people do ask me for advice, and to try and just be clear when I'm just telling them what I went through versus and they can take with it what they will. I'm describing it as advice. I think probably some of the best advice I got from Leanne was when I was trying to figure out hockey, and trying to, I had moments of doubt of, is this something I can keep doing? I actually went through in hockey in 2011. I was the coach at the time, cut me from the program. And so I thought my career was over then. But then a new coach came in, and the next year I was back in the fold. But I did a bit of thinking of like, do I want to get back into it? Because life was very busy. And Leanne essentially told me that, science will always be there, but you've got a chance to do something you love, and you should do what makes you happy and go and play hockey.
I think it is very exciting to find mentors who really who give you this piece of advice that science would be there and you go explore other things. Since you have seen science from, like, you are in Harvard Medical School right now, that is the topmost university, and you have played for Canadian team in Olympics. So what do you think is an intersection point of science and sports?
Yeah, so I think there's a lot, and I can touch on a few of the overlaps. I think one thing that for me at least is really true in both of those contexts that you mentioned, is always being aware of like how much everyone around you brings, like all the talented people around you, and how much you have to learn from everyone else. And with that, like, this sounds more like a sport thing, but I think it applies as well to science. But just the idea that you always keeping an eye on what you want to improve on. And so never feeling like you've made it. Like it's when I played hockey, I always had to keep in mind of, you got to draw confidence, in what you can do, but also know that, there's many things you can't do that you want to do. And so how do you keep improving? How do you keep learning? And recognize that everyone you meet is in the position to teach you something new and something cool. And so I think that's really important. I think that both, for me at least, both sport and science. One other thing is in sport and science, it's, especially in sport, or I think of it in sport first, and science, is taking a long-term goal and seeing what needs to be done to achieve that goal one day at a time. And so you can look at something like, we want to qualify for Rio in 3 years. And that's too far away to be tangible and to be actionable. And so rather than that, we can think about what can we do today to get us to that goal. And you break the big goal down into what do I need to do right now, and be in the moment in what needs to be done. And that's, as for sport, that's how I found the best way to achieve long-term goals was each moment. like living in the now. And I think for science, that's really important too. Once you have that long-term goal, if I want to answer this huge question, you have to break that down into what is the first question I need to answer, and what is the experiment I need to do today to do that.
Yeah, I think that is the most important thing. Like you have to be aware of the question you want to address further.
Do you have any advice on how one can go about selecting a PI?
In the vein of what I've said previously, I'll tell you what I did. And you can see that as my experience, and you could take from it what you will. And so I think there's a few things that I focused on. One is first the science, like what type of questions, what type of science would I do in a particular lab, and am I excited about those? Because that PhD is a long process, and if you're not excited about the science, it's going to be really hard. But if you are, then you can get through it. So that's number one. I think that the potential supervisor kind of interacts with trainees is really important. Is there a focus on mentoring? Is there support of the person as a whole rather than just the person at the bench? I think the broader group, so who your colleagues are going to be, like so much of your day to day are the other people in the lab, and I've been really fortunate to be in labs with great people who just make it fun and support each other. So those are kind of the main things. I would say the city does matter because you want to be in a place where you can do things outside of science. I think that's really important. So being in a place where whatever your hobbies or interests are, they can be met.
What do you think? How have PhD changed you in person?
That's a good question. Like I feel like my PhD was so formative. Like I went through so many big life experiences through it. It pushed me in all sorts of different ways. So I think it has doubtless changed me in many ways, and I think most of them I'm probably not aware of. When you're in it, it's hard to think back to where I was before and where I am now. One big thing that was part of my training and part of something that I think is how I like seeing science and seeing the world in terms of questions rather than facts. And so that's a big mindset change, I think, from my undergrad through as I started my graduate school. So questions rather than facts is probably one way. But I'm sure if you ask my wife, she'll give you many more.
Fragile Nucleosome is a brilliant initiative to promote science, and being a co-founder of Fragile Nucleosome, can you give us a few words about it for our young listeners?
Yeah. So this started during a year and a bit ago when much of the world was in lockdown from the pandemic. And basically a bunch of us were sitting at home with nothing to do. Twitter was full of the end of the world and was a bit discouraging. We wanted some place to just talk as scientists more about the science itself and like personal support and not so much of world news. And so this was Christine's idea and her initiative, and I was able to come on as a co-organizer and help with it. And it's an online community of chromatin and transcription and epigenetic scientists, a mixture of trainees and PIs, undergrads, and everyone else in between. And it's on one level a big support network. It's a place for scientific discussion, talks about the scientific endeavor. There's a journal club that comes out of it.
Motivation is key behind excellence. Was there any famous individual or scientist who inspired you in your journey?
And yes, the question is that's a great question. The question of who are my favorite scientists, who have I been and continue to be inspired by, and I think I have a long list. And if you ask me who's top of that list at any moment, it would change daily or hourly. But one of the people who I'm inspired by recently is Melissa Moore, who's an RNA biologist, had a long career as an academic, and is now the chief scientific officer at Moderna. And so she's now using basic science to really try and help people with these RNA medicines. including one of the COVID vaccines. And so I think she's just awesome. I'm really inspired by what she's been able to do, and it's stemming from this deep understanding of the basic biology of RNA, and then seeing how that can be used. And so as someone who does study basic biology, it's really cool to see.
That's fantastic.
Yeah. So managing time has always been a task. So how do you manage your time?
How do I manage time? I think the one word answer is poorly. I think maybe the one thing I try and do is I do try and keep either written out or mentally like a prioritized to-do list. And I try and make sure I'm spending as much time I can at the things on the top of that to-do list. So keep in mind what's really important that I do and make sure I'm getting to those.
Fitting an international hockey field around graduate studies is not a cup of tea for everyone. So how do you keep that work-life balance?
It's important to have things that you care about outside of science, and so there's things pulling you to that life in the work-life balance. And so for me, that's at times in sport, it's been relationships. It's been, wanting to get outside and go hiking. And then I find for myself, I need to make hard boundaries. Like sport was great because you can't miss practice. And so if I have practice at 7, I have to be out of the lab at 6 or whatever. And that's like a hard boundary. And once you have that hard boundary, it's amazing how science just fits within it. And if you don't, it can just kind of extend to wherever the loose boundary is. So I like to say that the time spent on research is kind of like a gas. It expands to fill the available vessel. And so trying to put in, and I'm not great at this, but trying to put in some hard boundaries and saying, okay, we're going for dinner at this time, and then sit down and figure out my day so that I'm done at that time is when I'm doing well what I do.
That's a beautiful comparison, by the way.
So everybody goes through failures, and everybody have different combat mechanisms to cope up with failures. Obviously, it doesn't come easy, but how do you deal with your failures? What are your combat mechanisms for failure?
Yeah, and so this is one area where I think sport has really helped me, because in sport there are so many failures on different levels. It can
Absolutely.
be within a game, in a moment, I might lose a one-on-one battle and the other team scores. You could lose a game, you could lose a tournament, we might not qualify. Like we didn't, one disappointment was the 2014 World Cup we didn't qualify for. It can be personal rejection, like I was cut from the team and I didn't make teams, I was cut from the program, like all those sorts of things happen. And so I've had plenty of experience of dealing with failure, which puts me in a great place for science. But I think there are a couple of things that I try and focus on. And so one thing which is hard, but I think even if you don't get all the way there, focusing on it makes it better, is disconnecting my sense of self-worth with how I do in science or sport. And so, if an experiment doesn't work, I can be upset about that because the experiment hasn't worked, but I don't want to see that as a reflection on who I am as a person, because that's a whole different level. And it's the same for sport. When I didn't make a team or I was cut, I had to see that as I'm disappointed because this is something I really wanted, but disconnecting that from my image of self-worth is really important. It's really hard to do, and I don't think I'm there yet, but I think reminding myself of that helps. And then I think the other, like I touched on outside endeavors, and so having things that I care about outside of science is really helpful. If things don't work, I can be excited about the hike we're going to go on, or I can be excited about a hockey game I'm playing and go and do that and forget about it, and that's really helpful. And then, I think the other thing with being an athlete is, I talk about goal and breaking that down to small steps. And so, we don't qualify for the World Cup. All right, what's the next goal? okay, it's for this next thing, you know, what to get there, what do we have to do? And okay, what do I have to do today? And so just kind of getting back into now and thinking, what can I focus on next is another way that I get through it. And it's the same now for science. If, you know, an experiment doesn't work or an hypothesis is wrong or we get scooped or whatever else, it's about, okay, what is the next goal and how can I try and get there? So those are some of the ways I deal with it. I think it's different for everybody, and the same. Even it's, for me, I find it's different in different times. Sometimes, you know, some of the things I said work, and sometimes they don't, and that's just it. You know, we're a different person as we come to these different experiences, and sometimes the rejections themselves or the failures hit us harder.
The application process to any good university can be quite a nerve-wracking task. Do you have any application tips for research aspirants?
The one thing I would say that I have heard from people, and I think is true, is if you are looking to apply to a specific school, and there are profs there in the department whose research someone is excited, if you're excited about it, it's worth emailing that prof saying you're interested in doing grad school. Make sure to include in there that, like, make sure to have read a couple of their papers and have something specific about their papers and their research, which is why you're emailing them. So they know that, oh, you know, this person has read my papers, they've got some interesting thoughts on the research. They're interested, they're emailing me not because I'm a name on a website, but because they care about my research. That's important, and you know, you should be emailing because you are excited about their research. So that those sort of setting those emails ahead of time and just saying, hey, I'm thinking of applying. It can, if you get in, it can turn into one of your rotations, which is great, and it can also give you a feel for the place.
You must have mentored a lot of young students while you were doing your PhD and now when you are doing your postdoc. So what do you think, what are the 3 best qualities that a mentor should have?
It's a tough question. What I try and focus on when I do mentor somebody is I try and understand them, understand what their goals are and kind of what gets them jazzed about science or doesn't. So I want to understand where they're coming from and what they want out of it, and how we can kind of have our goals align when we're working together. I try and empower them. That's the other thing. Like, I think it's and that's a bit of, like, I just know from myself, like, when I was empowered to be given a question and just trying to answer it, that's what got me excited. So I try and empower them in their projects. And I try and bring a level of excitement. Like, I think, and again, this is what I experienced when I started in the lab, is I was interacting with grad students who were excited about some of the research questions we were getting at, and I fed off that excitement.
Is there a particular book that you suggest people to read, and like that you suggest especially the students that you're mentoring to read?
So I don't have a particular, like, science book or, like, research
it can be anything
book.
It can be anything.
Yeah, I mean, I quite enjoy, like, I grew up reading a lot of Terry Pratchett, and so I like those books. And so if you want something outside of science that's just kind of fun and relaxing, and funny. Outside of that, it's not a book, but I would still put in a plug for the Fragile Nucleosome YouTube channel.
Right.
It's just an amazing collection of seminars, with so generous of all the speakers to share their work there, and to make it publicly available. And I think that's, you know, it's that sort of library of videos is such a resource. And so I would encourage people to take advantage of that.
Thank you so much. We'll end our interview here, and it was amazing to have you here. And thank you so much for accepting our request.
Great to hear. Hopefully. Thank you so much, both of you, for having me on. It's been really cool getting to chat with you both and hear a bit about the kitchen lab and the work you guys are doing. I think it's great. Keep it up. I watched your first video, and I'll be watching the rest.